code. Ribulose, bisphosphate, oxygenase. Yes, I was telling you that Rubisco, the same enzyme, ribulose, bisphosphate, carboxylase, oxygenase. Same enzyme acts as carboxylase. Ananya Thakur, my voice is cracking, so I think there is connectivity problem to your end. Everybody else is okay? Everybody else is okay? Is my voice clear to everybody? Or my voice is okay? But these days, Tadar Ani, my voice is not okay. So I think this is the problem from your side. Ananya, just check your connectivity and adjust at a place which is where the connectivity is better. Just bache. Okay. So yes, Ananya, just check the connectivity. Fine. Now, yesterday we were discussing photorespiration, and I told you the same enzyme, which is Rubisco. Or biphosphate, carboxylase, oxygenase. The same enzyme works as carboxylase as well as oxygenase. This depends on the condition. This also I told you, this depends on the condition where it will act as oxygenase and where it will act as carboxylase. And I told you in C3 cycle, it acts as carboxylase. This is C3 cycle which is going on, which we have done. We have done the complete C3 cycle, which is one of the part of the dark reaction, in which I told you that here, in this case, the Rubisco acts as carboxylase. Rubisco, it acts as carboxylase. Whereas I told you there are two conditions in which the Rubisco acts as oxygenase. The conditions are high temperature and high concentration of oxygen. So Rubisco here at this stage, it acts as oxygenase. The same enzyme can act as both carboxylase as well as oxygenase. So the conditions which are required when it acts as oxygenase are high temperature, and the second condition which is required is high O2 concentration. If these two things are there, then the Rubisco will act as oxygenase. And this is what I told you, that here in this condition, which is shown here, this row, this flow chart on this side, this shows that it is acting as oxygenase because see, it is combining with O2, and it combines with O2 only when it converts into oxygenase, or it is acting as oxygenase at high oxygen concentration. So children, I think it is very clear that why RUBP acts as oxygenase when the temperature is high, and when the oxygen concentration is also high, then it acts as oxygenase. Okay, now beta, we will just a minute, there is a, now in this case I was discussing this, what I have written on the board for you yesterday. I was discussing this, and in this, if you see, I have made 3 columns: the first column, the second column, and the third column. The first column is chloroplast, the second is peroxisome, and the third one is mitochondria. I have made the third one, mitochondria, smaller than the others because the written part was less, so I made it smaller than these two, and the written part was more in the chloroplast and peroxisome, so I made them bigger. Now here, if you see the RUBP, Rubisco is the enzyme, ribulose biphosphate is one of the reactants. There is difference. Rubisco is this is the enzyme, whereas if I say only RUBP, this is a substrate or a reactant. This is a reactant. Here, if you see RUBP at one time is combining with CO2. This we did in C3 cycle also, when RUBP combined with CO2. Okay, so when RUBP combines with CO2, the Rubisco, which is the enzyme which in the presence of Rubisco only this combination of RUBP and CO2 takes place. So when this RUBP combines with CO2, this is CO2 written here and this is RUBP written here, in the presence of RUBP and CO2, when they combine, this reaction takes place in the presence of Rubisco. And here, if you see, the concentration of oxygen is not high. So what happens? This RUBP acts as carboxylase. So then, as we discussed in C3 cycle, then PGA is formed, phosphoglyceric acid, then phosphoglyceraldehyde, and ultimately oxygen is given out, and at the same time some ATPs are utilized. This is biosynthetic pathway. C3 cycle is biosynthetic pathway in which whatever is formed in non-cyclic photophosphorylation, that is utilized, that is high energy compounds are utilized to form carbohydrates from CO2. This is what happens in dark reaction. So if CO2 concentration is high, it means if the temperature is optimum and if oxygen concentration is not low, the Rubisco acts as carboxylase and C3 cycle goes on. But if temperature is high, I told you, high temperature, when it acts as oxygenase, their temperature should be high and oxygen concentration should also be high. So at high temperature and high oxygen concentration, in that condition, the enzyme Rubisco acts as oxygenase, combines with oxygen, forms phosphoglycolate. This is still here, it is done in chloroplast. Then after the formation of phosphoglycolate, it gets converted into glycolate. This phosphoglycolate gets converted into glycolate, and this here only it will get converted into glycolate first. It gets converted into glycolate, and this glycolate then shifts to gets transferred to peroxisomes. When this glycolate gets transferred to peroxisome, there at peroxisome, this glycolate gets converted into glycine. Glycine is an amino acid, as you all know. Then after the conversion of glycine, the glycine enters the mitochondria, and when it enters the mitochondria, it gives out one amino group. So one amino group is given out. When the glycine gives out one amino group, it becomes serine, another type of amino acid. After giving out one amino group, the glycolate becomes serine, glycine becomes serine, and then this serine enters the peroxisomes again. This column indicates peroxisome. So the serine enters the peroxisome again. When it enters the peroxisome, it takes in that given out amino group. This amino group was given out here, that is taken up by serine, and then serine becomes glycine. So this way this serine and glycine circulation takes place in this area between mitochondria and peroxisome. One more thing which we have to keep in mind and which is very important. That very important thing is that I'm going to draw with the red color. This is very important here. This very important thing is, when this conversion of glycine to serine takes place, at that time CO2 is released in the air. CO2 is released in the air. All this is taking place in the presence of light, and then CO2 is released in the air. That is why the name photorespiration. Taking place in the presence of light, and what happens in respiration? CO2 is released, CO2 is given out here. Then after that, this serine enters the peroxisome, where this gets converted into glycine, and some of it gets converted into glycine again. So the circulation of serine glycine takes place. How it gets converted into glycine? By accepting the amino group which was given out by the glycine. It accepts the amino group, becomes glycine again. Okay. Then here most of it gets converted into pyruvate, then glycerate, and then this glycerate enters the C3 cycle at that state, PGA. It is accepted by phosphoglyceric acid, and then this enters the C3 cycle for the synthesis of glucose. This enters the C3 cycle. Now, this is how the complete photorespiration takes place, which results in the formation of CO2. Now, this photorespiration, very important. This is a wasteful process. This is a wasteful process. This causes no gain in any way. Why this is known as a wasteful process? And the plants, they do not want to have this process, and they some of the plants, they adapt some qualities, they undergo some sort of adaptation so as to prevent photorespiration. Why this photorespiration is known as a wasteful process? This is because of few reasons. Number one, the most important, CO2 is given out. If you see here, CO2 is liberated, is released. So this is one reason why photorespiration is a wasteful process. The second reason, which we can say that photorespiration is a wasteful process, because if you see in the complete process, nowhere any high energy compound has been formed. Neither, no biosynthesis has taken place. Like in C3 cycle also, no ATP, no NADP is formed. If you remember, I think you all remember now that in C3 cycle, NADP and ATP is utilized. You remember this, beta? How many ATPs are utilized in C3 cycle? How many ATPs are utilized in C3 cycle? Who will tell me that? Yes, Yashika, how many ATPs? Tadar is saying 6 ATPs are utilized. No, beta, not 6, more than 6. Those were more than 6. Ananya Thakur 3 is absolutely wrong. No, another one is also 3. How can you say that in C3 cycle only 3 ATPs are utilized? Yash, no, not even 12, Yashika. And second attempt is also not correct. No. If you remember, in one C3 cycle, yes, who's saying this? Priya is saying 18. That's right. 18 ATPs are utilized. I told you, see, 3 were formed on one side and 6 were formed on one side. 9 when 3 carbons were used. Here, one on this side, when I made the cycle on this side, 3 were formed, 6 were formed, and this side by the end in the regeneration process, in the regeneration 3 were formed. So total was 9. 9 in case when 3 carbons are used. So when 6 carbons, which is glucose C6, so in that case 18 ATPs. 18 ATPs are utilized, and how many NADPs are, and this calculation, this balance sheet of utilization of ATP is very important. The questions are asked based on this only. Now, if you remember, can you tell me how many NADPH are utilized in the C3 cycle? 12. Yes, Tadarani. Yes, Priya. It's 12. 12 ATPs are utilized. When we made the cycle, we made for 3 carbon, and in that we wrote 6 ATP. So 6 carbons, 12 NADPH. Okay. So here, in that case, in C3 cycle, which you remember, in C3 cycle, 18 ATPs are utilized and 12 NADPH are utilized. Okay. You can say that C3 cycle is also a wasteful process, because see, so many ATPs and NADPH are utilized. Why are you saying that C3 cycle the plants are accepting, they don't they don't carry on any adaptation to prevent that cycle? C3 cycle is important because it is we cannot call it a wasteful process, because in C3 cycle biosynthesis takes place. After utilizing 18 ATPs and 12 NADPH, something is formed, something is synthesized. And what is that important thing? That important thing is glucose, C6H12O6. So it is a biosynthetic phase. Of course, even if you cook food, you have to burn some CNG, you have to burn wood. So this is a biosynthetic phase in which glucose is formed by utilizing these ATPs and NADPH. So we cannot say that C3 is a wasteful process. If C3 will not take place, we will not get the food. So that is a biosynthetic phase. That is a biosynthetic process. But this is a wasteful process because see, what is happening? CO2 is liberated. In that case, no CO2 is liberated. CO2 is utilized to make food, to make glucose. Here, CO2 is liberated. First wrong thing which is happening, which is against the nature, which is harming the nature. That first thing is CO2 is liberated. And the second thing is no high energy compounds are formed. No high energy compounds are formed. Which high energy compounds are we talking about here? ATP, NADPH. Nothing like this formation takes place. No such formation takes place. So that is why we call that this is a wasteful process. Okay, and the plant they develop some adaptations to prevent this, that we are going to learn today. Okay, now here, children, anything in this topic, photorespiration, if you could not understand or you want me to repeat, you can please ask, because everything, these processes which I am telling you these days, C3, photorespiration, non-cyclic, cyclic, and then now C4, very, very important processes, very, very important processes. And, you know, even in NEET, many questions are asked from this. So just be very clear about these processes. If you are not able to understand anything from this, which we have done just now, photorespiration, just ask me. If you want me to repeat, I'll repeat, but you should learn. You should learn. These things should be on your tips. So any query, any repetition, if you want, just you can then tell me, quick Peta, I'm waiting for your response. Anything in the process photorespiration which you want me to repeat, or any query, anything. Even if you don't understand the complete process, I'll repeat, no problem. But you should know it properly. It should be thorough and clear. Yes, Bache, I want your response. Yes. Yes, beta. Not getting any response. Yes, Yashika. Sure. I'll be asking questions. Okay. Everybody is saying it is clear. So I think one session I'll keep for questions, and I'll ask questions which will be based on some of the processes. Okay. So here we finish up with the photorespiration, and we start with the next process, which is seafood cycle. So we start with the next topic, which is C4 cycle. This is also known as hatch and slack pathway, because this is the name of the person who discovered this cycle. So C4 pathway, this is also known as. Now, where this pathway is seen and why this pathway is developed by some plants. So this is seen in the plants, like examples I'll give you first. Where is it seen? Like sugarcane. It is seen in sugarcane. It is seen in sorghum. It is seen in maize. It is also seen in euphorbia. So this is seen in these plants. That means these plants follow, more are there. This is etc. It's not that some more plants are not there. More plants are there. I have written few. So few names I have given you as example. So these plants with the example of which I have given, some of the examples I have given, not all. These plants, they follow C4 cycle, which is also known as hatch and slack pathway. Now, what is this C4 cycle? C4 cycle basically is an adaptation shown by some plants to avoid photorespiration. So the plants which show C4 cycle, this C4 cycle is basically the plants, the adaptations developed by some plants to avoid photorespiration. Okay, one thing I forgot to tell you of photorespiration. When I wrote this photorespiration, I remembered. You know, the picture which I just erased, in that one compound was formed, and that compound I circled, and the name, it was formed in peroxisome, and the name was glycolate. If you remember, I made 3 boxes like this. This was chloroplast, this was peroxisome, and this was mitochondria. Here, if you remember, from here I made that glycolate. Here what is synthesized from phosphoglycolate is glycolate. Here I wrote glycolate, if you remember. This glycolate is a 2-carbon compound. This I missed, that's why I'm telling you. This is a 2-carbon compound, and because this glycolate is a 2-carbon compound, this process of photorespiration is also known as, sometimes known as C2 cycle. This process of photorespiration is sometimes is also known as C2 cycle, because one of the compound which is known as glycolate, which is formed, it's a 2-carbon compound. That is why this is also known as C2 cycle. So this point I forgot to tell you. What else this photorespiration is known as? What else this photorespiration is known as? Anybody? What else is this photorespiration known as, beta? Anybody who can answer this question? C2 cycle I have just written, Tadarani. I have just written C2 cycle. One more name I wrote when I was teaching you with red color. This photorespiration is a, yes, what type of process? What type of process? Yes, what type of process? Wasteful, not product. Not product, it's a process. This photorespiration is a wasteful process. And why do we call it a wasteful process? Why photorespiration is said to be a wasteful process? Why photorespiration or C2 cycle is considered as a wasteful process? Any point if anybody can give? Why this is considered as a wasteful process, beta? Yes? Yes, I want your response. Why this photorespiration? Yes, CO2 is liberated and no energy is formed. Yes, Tadarani. Yes, I would like to see your face sometimes, because I haven't seen you. Rest of the students I have seen. You are a new admission. I haven't seen you. So just put your photograph on your ID sometimes. Okay. So photorespiration is, yes. Photorespiration is a wasteful process because, okay, Tadar. Photorespiration is a wasteful process because no high energy compounds are formed, no ATP, no NADPH, and at the same time CO2 is liberated. That is why it is a wasteful process. Just now I told you from a few minutes back. Okay, that's why it is wasteful process, and most of the plants, they want to avoid this. And these C4 plants, they have developed some adaptations to avoid this process of photorespiration. These adaptations are seen in the plants which show C4 cycle. Now what happens? How they have, what adaptations they have developed to avoid the process of photorespiration. The first adaptation which they have developed is that they have dimorphic. Now I'll tell you about the adaptations which have been developed by these plants to avoid the process of photorespiration. The first process is they have dimorphic chloroplasts. They have dimorphic chloroplast. And the second thing is the presence of Kranz anatomy. Presence of Kranz anatomy. These two adaptations are seen in C4 plants to avoid photorespiration. Now, what is this dimorphic chloroplast? What is the meaning? What is the literal meaning of dimorphic? Anybody who can tell me? Dimorphic. What is the meaning of dimorphic? Yes? Yes, beta? Anybody who can tell me the meaning of dimorphic? When I say they have dimorphic chloroplast, what is the meaning of dimorphic? Anybody who can tell me the meaning of dimorphic? Break the word. Dimorphic. Yes? Yes means two. Yes, di means two. Dimorphic and morphic. Yes? Yes, bache? Dimorphic. What is morphic? Yes. Anybody? Amrita, you are sitting quietly today. Yashika Bhardwaj. Priya. Mannat. Lalit. Yes. No answers? Yes, beta. No answers from any of you? Dimorphic. Yes or no? Yes or no? I want your response. Yes or no? Yes, bache? No, I'm not getting any response. Two different forms. Yes, Amrita says two different forms. Yes, this is right. Two different forms. Tadarani also says yes. That means two different type of morphology are seen in chloroplasts. Chloroplasts are of two types. They are of two different morphology. Okay. And those two types of chloroplasts which are seen in them are granule, and agranule. Wherever we use the word a, that means without. Granule means where the grana are present. Agranule means where the granas are absent. This means simply means this. Now granas are present, that means they will have a structure like this. If this is one chloroplast, double membrane, we will have stakes of thylakoids. Okay, like this, we have thylakoids arranged in stakes. Thylakoids, which are joined by the stroma lamella, connected by stroma lamella. So what we have in granule, granas are present. But in agranule, the granas are absent. That means if we have this chloroplast, which is double layered, if we have this chloroplast, it will just have stroma lamella. This is one stroma lamella, this is another stroma lamella, this is another stroma lamella. Just stroma lamella are present. This is stroma lamella. And here both, these are stroma lamella and these are grana. And each grana is made up of many thylakoids which are put in stakes, one above another. So this is the difference between granule and agranule. That means in agranule, the grana are absent, whereas in another grana are present. Now one more difference between the two. The granule chloroplast, that means the chloroplast in which grana are present, these are present in mesophyll cells. So this was first difference which I told you. The grana are present, grana are absent. The second difference is present in mesophyll cells. Mesophyll cells. Whereas these ones, that means agranules, these are present in bundle sheath cells. Now you will say, what are these bundle sheath cells? These are present in bundle sheath cells. Now you would like to know what are these bundle sheath cells. For this, I'll have to make a diagram which you learned in class 10th. And before also, when I was telling you site of photosynthesis, at that time also I told you that diagram. See, we have a upper epidermis. This is upper epidermis. And this upper epidermis has cuticle. This has cuticle. And then we have lower epidermis. And in the lower epidermis, we have this stomatas. So we have stomatas in the lower epidermis. And what do we have in between? In between, we have here mesophyll. Mesophyll is divided into two types of parenchyma, palisade and spongy, if you remember. So palisade,